Good morning, everyone. Thank you, Ralph. That was very inspirational. I'm ready to go back and do another postdoc. Um, it's my pleasure today to introduce the first speaker. Dr. Galia is an assistant professor of pathology and a secondary appointment in immunology. She joined us at the end of 2023. She got her PhD from the Weizmann Institute in Israel and then joined the laboratory of Kate Fitzgerald at the UMass Chan Medical School in Worcester, Mass, where she really explored a variety of topics, most of which she's carried forward to her studies here. She's been a recipient of multiple grants from the Crohn's and Colitis Foundation and is a recent recipient of the NIAID Directors Award for New Innovative Science, a so-called DP2. Her science focuses on understanding the role in both health and disease, because we are a pathology department, of lincRNAs. These are long, non-coding RNAs, which are found throughout the body. Every cell expresses these, and they're probably the poorest characterized nucleic acids anywhere. She has shown that tissue-specific lincRNAs are instrumental in the function and the normal function and in pathology found in the colon. Look forward to hearing her talk about how these lincRNAs interact with mitochondrial function and preserve the ability of cells to make ATP. Faraz?
Okay, thank you, Jim, for the nice introduction, and thank you for the opportunity to present my science. This is really a pleasure. So my name is Galia. I'm assistant professor at the Department of Pathology and Immunology, and our lab is mainly focused on identifying a role for long non-coding RNAs, also known as lincRNAs, in intestinal health. So we are mainly focused on ulcerative colitis, which is a form of inflammatory bowel disease, which are characterized in ulcers and inflammation in the colon. And this is really a multifactorial disease, which in addition to the common symptoms that we are all well aware, including rectal bleeding and abdominal pain, it actually affects many other areas in our body. So this is really a global burden as it affects million people worldwide. And what I want to show here in this graph is that actually the number of patients diagnosed with ulcerative colitis is increasing, have we here, both in men and women. And treatment are a combination of anti-inflammatory immunomodulators and sometimes even surgery, which is correlated with the disease severity. But what I really want to emphasize here is that the current treatment actually only address the symptoms and maintain remission, and there is no really cure for ulcerative colitis, which really emphasize the need to identify novel regulatory factors that are playing a role in the pathogenesis of the disease. So looking on our human genome, we now know that the majority of our genome is actually transcribed as non-coding RNAs, and most of these non-coding RNAs are lincRNAs. LincRNAs are transcripts that are longer than 200 nucleotides. They have similar features to messenger RNA, so the majority of them are transcribed by RNA polymerase II. They are spliced, capped, and polyadenylated, but they lack protein coding potential. So they actually function as RNA molecules. And with regards to ulcerative colitis, we now know that the majority of the IBD-associated risk loci are actually located in the non-coding genomic region, mostly in the lincRNA. So it's really emphasized the importance of these genomic regions to the development of the disease. So this graph is just to kind of like summarize what we already know regarding lincRNAs in ulcerative colitis. These are really emerged molecules that are studied well in this disease, and they've been identified as regulatory molecules that regulate epithelial barrier function and mucosal immunity. So what I want to show in this graph is that actually when people have started to work on lincRNAs, they mainly focus on the function in the nucleus as chromatin modulators, regulate chromatin modifications. And with the years, we came to know that link RNAs can actually also translocate to the cytoplasm and act through various functions. So they actually regulate almost every biological process that we can think in the cells and regulate protein function and signaling. So what our lab is focused on is on a unique subset of link RNAs that can translocate from the nucleus to different organelles in our cells and regulate organelles function and immunometabolism, and thereby affecting colon health. So to address this, we first profile for link RNAs in the colon. So we have collected samples from mice, healthy mice, and mice that challenged with different models of colitis, as well as hundreds of biopsies from different areas of the colon, from healthy patient and patient with ulcerative colitis. And we started to profile these link RNAs, looking for link RNAs that are associated with the disease, that are differentially expressed in colitis, and that are conserved between mouse and human. So now what I want to emphasize with link RNAs is that are poorly annotated, and they are also very specific to a cell type and tissue. So it's very important to identify the exact transcript that express in the colon. So in order to do that, we performed Oxford nanopore sequencing. So this is a long-read RNA sequencing that enabled the detection of novel transcript in the colon. And when we combine the nanopore sequencing with our expression profiling, we were able to generate sequence library of all the link RNAs that are expressed in the colon with potential function in colitis. So we have identified by this method the link RNA HOXA11OS. And what I want to show in this graph, in blue, it's the annotated transcript that was reported before. And I want to emphasize the green transcript, which is actually a novel unannotated transcript that we identified by the nanopore sequencing. And what is unique for these link RNAs, it's only expressed in the colon. And even in the colon, it was only detected in the distal part. So this is an RNA scope analysis. It's a single RNA molecule hybridization assay. And each brown dot here is actually a single molecule of these link RNAs. So we can see here that it's only expressed in the distal part of the colon. And as we go to the proximal area, the detection is decreased. So what we found is that it's highly conserved between mouse and human, with 80% sequence conservation between the different transcripts. And what was unique about this link RNA is that it's actually in the myeloid cells in the colon was translocated to the mitochondria. So we can see it here by co-localization with mitochondrial ribosomal RNA, as well as a biochemical approach of isolating mitochondria for intestinal immune cells. And what we found is that this link RNAs actually interacts with a protein which is part of the complex 1 of the electron transfer chain, the NADH dehydrogenase. So this protein is part of the complex 1, which is coupled to the TCA cycle, and through the conversion of NADH to NAG, we then have the electron transfer chain that go through the different complexes, and in the end we have the production of the ATP, which is the main source of the energy of cells in our body. So then we generated mice that are deficient for this link RNA, and what we found is that when this link RNA was deficient, we have dysfunctional mitochondria. So we specifically have dysfunctional complex 1 activity, and this was associated by reduced conversion of NADH to NAD, elevated level of mitochondrial ROS, and reduced production of ATP in intestinal myeloid cells. And this was associated with elevated level of inflammation in the colon. When we challenged the mice with DSS to induce colitis, we saw that these mice are highly susceptible to colitis compared to wild-type litterman control. Looking on the mitochondria of the intestinal myeloid cells, we can really appreciate the phenotype of these mice. So comparing the wild type to the HOXA11 OS deficient mice, we see swollen mitochondria. We have complete loss of the crystal structure of the mitochondria in these myeloid cells, and we have swollen mitochondria with loss of density. And this was associated with a decrease of a mitochondrial mass and decrease membrane potential of mitochondria in these cells. So taken together, what we found here is a novel link RNA. So this was the first identification of a link RNA that can translocate to the mitochondria in the tissue microenvironment and really regulates the mitochondrial respiration in these cells. So under basal condition, HOXA11 OS traffic inside the mitochondria and interact with this protein of the complex 1. And when this link RNA is deficient, or during colitis when it's downregulated, we then have dysfunctional complex 1 activity, and this is associated with elevated level of mitochondrial ROS and inflammation in the colon. So our lab is now focusing on expanding on this finding. So we are focusing on 3 main studies. So the first study will be to identify novel link RNAs that can translocate to other organelles that are associated with the severity of ulcerative colitis. So we are focusing on the peroxisome, lipid droplets, and the ER, identifying link RNAs that can traffic into these organelles and really regulate immunometabolism. We're also interested in link RNAs that can translocate to the membrane contact site between organelles, because we now know that organelles work as a network. So we are interested in link RNAs that can really traffic to the site and regulate organelle communication and exchange of signals between these organelles. And another study that we have is the focus on link RNAs that can secrete by exosomes from different colonocytes, and how these exosomal link RNAs can really regulate communication between cells in the colon, and how this affects colon health. So we use diverse techniques for this to answer this question, including the MERSCOP, which is multiplex RNA hybridization assay. It's similar to the RNA scope, but we can actually probe for hundreds of RNA with a combination of protein to label different organelles, so we can then look for co-localization of different link RNA of interest with organelles within the colon microenvironment, as well as biochemical approaches and genetic mouse models of TAG mitochondria and peroxisomes. So with this I will end, and I would just want to first thank my postdoc mentor. So most of the HOXA11 OS study was done during my postdoc. So Kate Fitzgerald was my mentor, and the people in the lab who helped me. Of course, the foundations that supported this grant, and Jensen, which collaborated with us on this project. And Jorge and Noah from UPenn, who helped us with the strategy to knock out HOXA11 OS in these mice. And my current lab, Shaika, he's a postdoc in our lab, and Nolan, a research assistant, which currently work on these models in our lab, and of course the funding agency. And thank you for listening, and I will take questions if any.